Okay, so welcome to this next video in which we're discussing nitric oxide as the endothelium dependent hyperpolarization factor. Okay, right, so we have so far seen how Chen et al. And Taylor et al. They seem to show that nitric oxide was not involved in endothelium-dependent hyperpolarization. We are now discussing what Taylor et al. In 1990 did, which seems to contradict that and show that nitric oxide does cause endothelium-dependent hyperpolarization. Okay, so basically, I've been explaining to you how if you pre-treat a blood vessel with noradrenaline or norepinephrine, as it's called in the States, it's going to result in both contraction and depolarization of the electrical potential difference across the plasma membrane. And this is both of these are caused by the release of calcium from the SR into the cytoplasm of the cell. That influx of calcium causes contraction, and also because it carries a positive charge, it's going to raise the electrical potential in the intracellular compartment. And if we can assume, therefore, that the electrical potential of the extracellular compartment remains the same, then the amount by which the intracellular compartment's electrical potential is lower than the extracellular electrical potential, i.e. the electrical potential difference, is going to be smaller. It's going to become smaller because you're raising the electrical potential intracellularly. So this number that was initially negative 60, which meant that the intracellular electrical potential was 60 millivolts lower than the extracellular electrical potential, this is going to become less negative. It's going to maybe go up to negative 50 millivolts. And this is known as depolarization. Okay, right. So this is what Taylor et al. Were using. They were using blood vessels which they had constricted with noradrenaline. Now, potentially, I don't know the details, but potentially Chen et al. And Taylor et al. Were not using pre-constricted vessels. They may have been using perfectly relaxed vessels. Okay, usually these experiments are done on pre-constricted vessels because it means that you can see, generally, a more pronounced relaxation effect. If your blood vessel's already relaxed, are you going to see any more relaxation? Potentially not. If your blood vessel's not already depolarized, are you going to see any more, any reduction in the, any hyperpolarization? Maybe not. Okay, any hyperpolarizing effect, I mean. Okay, right. So, let's now discuss what Taylor et al. Did then. What they did is they took blood vessels which were pre-constricted with noradrenaline. So these blood vessels are very tightly constricted, so all the smooth muscle cells of the blood vessel are contracting. They are causing the lumen of the blood vessel to constrict. Okay, so let's draw our little blood vessel out here. Okay, so here's our blood vessel with the endothelial cells sitting on the basement membrane here. Okay. Tunica media, this layer of smooth muscle cells around the endothelium here, and then a layer of connective tissue around that, known as tunica adventitia. Okay, so these are the layers of our blood vessel. And now what we're going to do, this is our pre-constricted blood vessel. What we're now going to do is treat this blood vessel with acetylcholine or carbachol again. So we're going to stimulate the M3 receptors that are on the endothelial cells with acetylcholine or carbachol, just like Bolton et al. Did in 1988, except that now our muscle's pre-treated, pre-constricted. Well, maybe, I don't know, maybe the Bolton one was as well. Okay, so we're going to treat with acetylcholine, or it could also be carbachol, CCH. Again, this is the M3 receptor that you've got on endothelial cells, the M3 muscarinic acetylcholine receptor. Okay, and what you find is that it causes, just as Bolton et al. Observed, it causes hyperpolarization of the muscle cells, so it has a hyperpolarizing effect on the electrical potential difference across the membrane of the muscle cell. So you see a hyperpolarizing effect. And then you also see a relaxation of the smooth muscle cell. Now, we would expect the relaxation because we know that acetylcholine or carbachol is going to stimulate the endothelial cells to produce nitric oxide, and the nitric oxide we know will cause relaxation of the muscle cells. It's the endothelium-derived relaxation factor, remember. Okay, so we expect it to cause relaxation, but we also see that it causes this hyperpolarizing effect that Bolton et al. Saw in 1984. Okay, so that's not new at the moment. So what is new is that if you block the production of nitric oxide in these endothelial cells using an inhibitor of the enzyme which produces the nitric oxide, namely an inhibitor of ENOS, endothelial nitric oxide synthase, also known as nitric oxide synthase 3. Okay, if you use an inhibitor of this, such as NMMA, okay, then basically what you see is an inhibition. You stop the relaxation and the hyperpolarizing effect of stimulating with acetylcholine. So let's discuss NMMA in a bit more detail. So ENOS is the endothelial nitric oxide synthase. So it is the enzyme that is present in endothelial cells, which upon stimulation of the endothelial cells with acetylcholine or carbachol, or in fact there are other receptors on the endothelial cells which can also stimulate this enzyme. In fact, all you need is calcium to elevate in the level of the in the intracellular compartment of these endothelial cells in order to get activation of the endothelial nitric oxide synthase. So many different receptors can cause calcium to go up in the endothelial cells and therefore activate the endothelial nitric oxide synthase. Now, when it becomes activated, what it's going to start doing is producing nitric oxide. Okay, so the substrate for this reaction, the substrate for the production of nitric oxide, is the amino acid L-arginine. Okay, and this is the basis of the inhibitors of this enzyme. Basically, LNMMA, which stands for NG monomethyl L-arginine. Okay, this is an analog of the structure of L-arginine, basically, and it will go into the active site of the enzyme and it will not undergo the correct reaction that L-arginine undergoes, and therefore it will just block L-arginine from going into the enzyme, and it will block the activity of the endothelial nitric oxide synthase. So let's have a look at the structure of L-arginine, and then let's see how you would modify NG monomethyl L-arginine to get this inhibitor of the ENOS enzyme. So this is LNMMA. Okay, right, so the structure of arginine then. If you have your amino group here, so L-arginine is an amino acid, so let's draw the core amino acid structure here. So here's the alpha carbon with the carboxylic acid group coming off it here, and also the amino group up there. And then the structure of L-arginine then has 3 methylene groups in. And because I'm too lazy to draw 3 methylene groups, there's a nice trick. You can just draw brackets around 1 methylene group and then put 3 down there, just to save yourself a little bit of work. Then you have a nitrogen atom next, and then after that a carbon, which has an amino group off it, and also a double bond to a nitrogen down here, which also has a hydrogen off it. Okay, so this is the structure of L-arginine. Now, if you want to turn this into LNMMA, or NG monomethyl L-arginine, what you do is you take one of the hydrogens off this amino group and instead replace it with a single methyl group. So what you'd have instead on the end of this molecule would be a structure like this. Here's this amino group, and instead now it would have this methyl group on here, and then you still have this double bond to the nitrogen and the hydrogen here. Okay, so that's the structure you'd have in LNMMA. Okay, whereas this is the native structure of L-arginine. So this is the basis of how this drug can be used to inhibit the ENOS enzyme. Okay, or NOS3. NOS3 is the more correct name for this enzyme. The reason being that it's actually found in a huge number of other places apart from just the endothelium. So that's why it's more correct now to call it nitric oxide synthase 3. Besides, regardless of that, it is very heavily expressed in endothelial cells, and in response to stimulation of these M3 muscarinic acetylcholine receptors on the surface of the endothelial cells, calcium will go up within the cytoplasm of the endothelial cells, and that will activate ENOS. So I will just put that, you'll get calcium going up, and that will activate the ENOS enzyme via calmodulin binding to the ENOS enzyme. Okay, and the ENOS will start producing nitric oxide, and nitric oxide will go through to the smooth muscle cells, and we know it causes relaxation. What's new to us is that maybe it causes this hyperpolarizing effect. Okay, so when we inhibit the production of ENOS and then try this experiment again, i.e. we stimulate with acetylcholine or carbachol, then again the calcium goes up, but now ENOS won't be activated, so you won't produce nitric oxide. Then what we'd expect to see is the relaxation gone. However, we wouldn't expect to see a loss of the hyperpolarizing effect, because remember, Chen et al., he got rid of the nitric oxide by flooding the endothelial cells with hemoglobin, which was mopping up all the nitric oxide, and he didn't see that the hyperpolarization was blocked. However, Tar et al. Did see a block of this hyperpolarizing effect. Okay, so they saw that the reduction, the more extremizing, well, the hyperpolarizing effect, I can't think of any other way of saying it, of acetylcholine stimulation of the endothelial cells on the electrical potential difference across the membrane of the smooth muscle cell, i.e. the tendency of this to be made more negative again. So, for instance, when we stimulate the endothelium-dependent hyperpolarization in these pre-contracted and therefore pre-depolarized smooth muscle cells, maybe it'll go back down to -60. In this case, the hyperpolarizing effect would repolarize the membrane. So when you are correcting a depolarization, it's called repolarization. When you go beyond, maybe to -70 millivolts, beyond the normal resting membrane potential across the cell membrane, that would be a hyperpolarization. However, a hyperpolarizing effect just means taking the electrical potential difference across the membrane to more negative levels, basically. Okay, so whether the hyperpolarizing effect is achieving repolarization or hyperpolarization is immaterial. Okay, right, so this appears to show that nitric oxide is important, since if we apply this nitric oxide synthase inhibitor, then you stop the hyperpolarizing effect that's triggered when we stimulate these endothelial cells with acetylcholine or carbachol. Okay, right, so Tar et al. Didn't stop there. They went on and did more experiments. So they basically did this same experiment as Taylor. They said, okay, if nitric oxide is important, then let's take the nitrovasodilators. So they took the nitrovasodilator, SMP, or sodium nitroprusside, okay, and they also took another vasodilator. Wait a second, let me try and squeeze this in. Sodium nitroprusside there. They also took another nitrovasodilator, which it has this acronym GTN, which stands for glyceryl trinitrate. Okay, so both of these drugs are compounds which, if they go into the blood, they will undergo chemical reactions, and in that chemical reaction they'll produce nitric oxide. So if we put either sodium nitroprusside or glyceryl trinitrate into our blood vessel, what's going to happen is it's going to produce nitric oxide, and that nitric oxide will be produced independently of stimulating the endothelial cells. So if nitric oxide is producing this repolarization of the electrical potential difference across these pre-contracted smooth muscle cells, then these nitrovasodilators, as they are, should cause this repolarization effect, and we know they'll cause the relaxation effect because that's mediated by nitric oxide, and we know that. Okay, so that's what they did. They used these two nitrovasodilators, and they found that they were capable of repolarizing the pre-contracted smooth muscle cells. So this seems to be in blatant contradiction to what Taylor et al. Showed in 1988. What we can say, however, is that firstly, Taylor et al. And Taylor et al. Were working in different species. They were also working on different arteries, and finally, the Taylor et al. Group, the Tar et al. Group, were using pre-constricted blood vessels, and I don't know what Taylor et al. Were using. They may have been using relaxed blood vessels. Okay, so it does seem that nitric oxide does have some sort of an effect as an endothelium-dependent hyperpolarization factor. And we'll continue our discussion further by looking at experiments that were done by Parkinson et al. In the next video.